So this is part 3 of the chapter 4 on cell structure and function, and just running down the list of eukaryotic landmarks, organelles mostly, but not only. Nucleus, cytoskeleton, mitochondria, lysosomes, the endomembrane system, which is the rough and smooth endoplasmic reticula, the Golgi apparatus, and vesicles. We have the cell wall, vacuoles, and chloroplasts. Let's go ahead and delve in. Starting out with the nucleus. Remember that eukaryotic cells are defined by having a nucleus, although strangely and interestingly, some of your cells will get rid of their nucleus as part of a normal process, specifically the red blood cells, which literally lack a nucleus at the end of the process, but they have it as they're going through the developmental process. So the nucleus has two main jobs. It's going to act as the genetic control center of the cell. What that means is it's going to tell the cell everything that the cell needs to do in order to stay alive. It's the general, right? It's telling the troops what needs to be done. It also stores hereditary information. When cells divide or when you have offspring, the information that's contained in that nucleus is passed on to the new cells or passed on to the offspring. So hereditary information. That's what the job of the nucleus is. It's very prominent. It's the largest organelle in the cell. And so that makes sense that we learn a little bit more details about this nucleus. Now, this is the nucleus. I'm going to go back a second to this slide here and remind you that it is a very big, but it's not the entire cell, right? It's a very big structure in that cell. And it also has this little center right here. And this center is called the nucleolus. A lot of people misidentify on tests and quizzes. They misidentify the nucleus versus the nucleolus. The nucleolus is the center of the nucleus. The job of the nucleolus is to make ribosomes. Do you remember ribosomes? We talked about this when we were first talking about the structure of prokaryotic cells. Ribosomes make proteins. And so, but ribosomes have to be made somewhere themselves. They're being made in the nucleolus of the nucleus. Next is the chromatin or chromosomes. Sorry about that little typo there. We'll look over here. Chromatin or chromosomes, same thing. The reason that it has a couple different names is going to become apparent when we start talking about cell division, because in cell division we're going to learn that we sometimes call the DNA chromosomes and we sometimes call the DNA chromatin, depends on how it looks. But that in this structure right here where you can barely see any of that, those little squiggly lines, we would call those chromatin. If you can start to see them very clearly, they'd be called chromosomes. But then again, they're always called chromosomes, so it gets a little confusing. One of the harder terms to learn about in biology is chromosomes versus chromatin. The second to last structure that we're going to talk about is the plasma membrane here. We call this the nuclear membrane. It's made up of phospholipids, but if you look here really carefully, it's actually a double phospholipid bilayer. Each of these thin layers here is a phospholipid bilayer. Remember what the phospholipid bilayer looked like? Just a real quick, yeah, we can find it on this page. This is the phospholipid bilayer right here, the two layers, hydrophilic, hydrophobic, and so forth. But it's a double layer here with a space in between. And so you can see here, there's this little gap right here. And so it's a really, really thick bilayer. It's a double bilayer. And the only way that things can move in and out of this nucleus are through these structures called pores. These are also made up of protein, but they don't really distinguish. They're not like transport proteins that say, hey, only you can go through. They just let anything in and out of the cell. And this is how the inside of the nucleus communicates with the cytoplasm that's out here. So our pores. The next one we're going to talk about is the cytoskeleton, and this is the scaffolding that makes up the cell. You can see it here, all these fibers that run through the cytoplasm, or the cartoon version of it here with these sticks and these little squiggles right there, squiggles and sticks. Cytoskeleton. Three main functions of the cytoskeleton include inner scaffolding, also providing shape to that cell, providing support for that cell. Very cool, the cytoskeleton acts as like a superhighway in the cell. So if things need to be moved from one area of the cell to the next, maybe you've got a vesicle that needs to be transported out of the cell, it's going to walk along this cytoskeleton. And finally, if the cytoskeleton is on the outside, and if that outside cytoskeleton can wiggle, you might have cilia, or cilia over here, or flagella. Remember that cilia are not the same thing as pili. Cilia help with things moving along the surface of the cell or help a cell move itself, and pili are like Velcro. They hold on to things. So they look similar, but they are quite different. All right, there's your cytoskeleton. The next one that we're going to talk about is the mitochondria. And mitochondria, you've heard this before, it's the powerhouse of the cell. I don't really like that nickname to use in conversation, but you've always heard it, so I'm going to use it for now. What I like to think about is that mitochondria take food energy and convert it into ATP energy. Remember, ATP is the type of energy that all processes within your cell uses. And you eat food, literally, for the most part, you're eating food so that you can make ATP. You need other nutrients too. You need to have building blocks too, but you got to make that ATP, and that ATP is being made in the mitochondria. So just some quick details here. It's got that double membrane. It's got DNA kind of floating all throughout there. It can make its own ribosomes, and it makes ATP. I like this little graph. The graph here tells you how many different types, numbers of mitochondria are found in various types of cells. So your liver cell has over 2,000, 2,500 mitochondria in every single cell. That must mean that liver cells need a lot of energy, and they do. They have a huge job in your body to detoxify everything and to make sure your body's moving, acting smoothly. The next is skeletal muscle at about 1,200 mitochondria per cell. That's a lot, and you use your skeletal muscles to move things, to walk, to write. If you're writing right now, you're using your skeletal muscles, so you need a lot of energy for that. White blood cells have a little bit less, and very interestingly, red blood cells have 0 mitochondria. Red blood cells are very passive in what they do. They just grab onto oxygen and release oxygen, and grab onto oxygen and release oxygen. That's pretty much all they're doing. So they don't really need any energy to do this grabbing on releasing, because it's all very passive. And oxygen is moving across the plasma membrane in a simple diffusion across its concentration gradient, and when it gets to your cells in the rest of your body, it lets go of that oxygen for the same exact reason. So no ATP going on there. Next one is the lysosome. And lysosomes have the nickname as being the cell's garbage disposal, which anytime your body breaks down anything, there's waste products. And the lysosomes are there to help with breaking that stuff down. In fact, sometimes maybe your cells are producing too many mitochondria, or those mitochondria become defective and no longer working. It's the lysosomes that take care of that. They clean everything up, make sure the cells are running well. Garbage disposal. But there is a disease associated with lysosomes called Tay-Sachs, and Tay-Sachs is the inability to make proper lysosomes. So lysosome grabs onto an organelle, but it can't digest it. So what you end up having is all of these molecules, all of this junk building up inside your cells, and you can't function when you are surrounded by garbage, by waste product. You can't function. And so what happens is, well, it's inevitable and it's unfortunate, but death. You literally cannot live if your body is filled with things that it needs to be getting rid of, waste products. So Tay-Sachs is a genetic disorder that often kills children before the age of 2. There is a slightly different version of it that's found in adults, but that's also deadly. They don't live much past 20 or 30. So the lysosome is actually a very important structure. We're moving on now to the endomembrane system. And remember, the endomembrane system is made up of 4 structures. I have 3 of them circled here for you. Adding the 4th one is the vesicle, which I'll give you some details on in a second. So in general, the endomembrane system has a couple of big functions. Number 1, it's going to make and it's going to modify molecules that need to be outside of the cell. So if you're making, again, we're going with adrenaline because we've already talked about it. If you need to make adrenaline, you're going to make that inside the endomembrane system. And then you're going to export that outside of the cell so that it can talk to other cells. The second function is to break down toxic chemicals and cellular byproducts. We already talked about lysosomes doing that a bit, and there are some parts of the endomembrane system that will also help with breaking down toxic, just the toxic ones, not the everyday trash. And so we'll get into the details there. But again, the parts are the rough endoplasmic reticulum with the dots on it, the smooth endoplasmic reticulum, it kind of looks like coral, and the Golgi apparatus, and that kind of looks like, bars on your cell phone, like how many bars you got. So let's get into the rough endoplasmic reticulum first. The rough endoplasmic reticulum, or the rough ER, is covered with ribosomes. And we already learned that ribosomes make proteins. So, 3 guesses about what the rough endoplasmic reticulum does. It's making proteins. And so, but remember, it's not making all the proteins inside your cell. It's just making the proteins that need to be exported from the cell to the rest of the body. Hold on a sec. Sorry about that. I just had somebody knock on my door. Anyway, we were talking about the rough endoplasmic reticulum and how it exports proteins to the outside of the cell. The next part of the endoplasmic reticulum that we're going to look at is the smooth endoplasmic reticulum. Notice that there are no ribosomes on there. It is smooth, and it has a couple of really important functions. Number 1, it's going to make lipids, like fatty acids, like phospholipids, like cholesterol or other steroids. That's what its job is, to make fats. The next really important function is that it's doing a lot of detoxification. So anytime you drink alcohol or take drugs, and I'm not talking about drugs, I'm not talking about just illegal drugs, I'm talking about any kind of drug that you take into your body, it's going to detoxify that. So you don't really need to go on a cleanse diet, right, to detoxify your body. Your smooth endoplasmic reticulum, which is found in every cell of your body, it's doing that. You actually do find a lot more smooth endoplasmic reticulum in your liver because the main job of your liver is to detoxify. So that's the smooth endoplasmic reticulum, or the smooth ER. Besides making lipids, it does make some carbohydrates, like the ones that we saw as the fingerprint on the outside of the cell. Remember that anything made inside the endoplasmic reticulum, that's mostly for export outside of the cell. So just a little bit more on that smooth ER, because it is fascinating. If you take a lot of drugs, and again, it doesn't have to, we're not talking about illegal drugs here, we're talking about any kind of drug, from antibiotics to heroin, any form of those. Anytime you take a lot of a drug, the smooth ER builds up. You get more and more and more smooth ER in your cells because it's trying to detoxify. So what happens is you get less of a long-lasting effect of the drug. Again, from antibiotics to heroin, that drug lasts less and less long. Sometimes people who who have been on this cycle like of heroin, where they build up and they build up and build up their ability to withstand heroin because their smoothie R is building up and building up and building up, and then they get clean for a while, and then they go back to taking drugs, and they take the same amount that they were used to taking before, but the cells have gotten rid of some of their smoothie R. It's not as necessary. And so what happens is you get overdoses way more often after you've been clean for a while than if you have been taking drugs for a while. Just an interesting, sad, really tragic, actually, side effect of this part of the body's functioning, of detoxification, building up. It's not, the word's not immunity, but building up resistance to able to take large amounts, so you can take more and more and more to have the same effect. The third part of the endomembrane system is the Golgi apparatus, and the Golgi has a really interesting function. It's like the final the final processing. So when something's made in the rough endoplasmic reticulum or the smoothie R, it's usually sent to the Golgi, and the Golgi will make sure everything's in working order. And then it's the Golgi that will send the material to the plasma membrane for export. Let's take a look at what that looks like. Oh, and by the way, remember I told you that transport vesicles were part of the endomembrane system, and then you see it right there. So let's take a look at how it all works together. You've got the rough endoplasmic reticulum, the smooth endoplasmic reticulum. This is making proteins, this is making lipids and carbohydrates, and they all get pinched off into these little vesicles called transport vesicles, and they're transported along the cytoskeleton, which it's not drawn here, but kind of use your imagination. Drawn along the cytoskeleton they get to the Golgi apparatus. When they're in the Golgi apparatus, they will go through this process of being modified and made into the proper form, and then that pinches off again, so we got another transport vesicle, and that transport vesicle takes it to the plasma membrane, in which case we get rid of that substance because someplace else in your body needs it. So vesicles, all of these four things work together in the endomembrane system. There's like this one exception though that I want to talk to you about, and that is lysosomes, because lysosomes go through this whole process but then don't go here. They just kind of hang out inside the plasma membrane, inside their little vesicle. Lysosomes. All right, so we've actually covered every part of structures that are found in both plants and animal cells. And I'm going to spend the rest of this time talking just about the structures found in plant cells. So the first structure we're going to talk about is the cell wall. And the cell wall is this very thick covering right here. There's the plasma membrane. And so the cell wall sits outside of the plasma membrane. And I have this question here. It's kind of rhetorical. I'm going to answer it for you, but I want you to think about it for a second. Is the cell wall, which is found here, is that inside of the cell or outside of the cell? And it's one or the other. It's either inside or outside. And hopefully you recognize that it is actually outside of the cell, because the plasma membrane is what divides inside from outside. And so if that's dividing inside from outside and all of this is inside, then the cell wall must be on the outside. If you look at a regular plant cell, like a live shot of a plant cell, what you're seeing here, this really thick part, that's all cell wall. It has a plasma membrane, but remember the plasma membrane is much thinner, and so it's really, really, really hard to see the plasma membrane. Maybe you can see a little bit of it right there where my mouse is pointing. What are the functions of the plant cell wall? Number one, these are very similar, by the way, to what we talked about with the cell wall with the prokaryotes. They give strength to the cell. They provide structure to the cell. For plants, you do have an additional function, and that is that it helps with water resistance. all this means is that this cell will not explode if it's exposed to too much water, because that wall will protect it. That's very different from an animal cell. Animal cells can, in fact, explode if they're surrounded by pure water for too long. So interesting to think about. And then finally, the cell wall does provide some protection against being eaten. We talked about that with the prokaryotes. Things that might want to eat plants maybe might not be able to if they have this really tough cell wall. The next is the vacuole. Remember, we said that vacuoles can be found in animal cells, but they're temporary. They're permanent structures in plant cells. So, and they're fairly large structures. Like, check this out. This is the vacuole inside this whole cell. It can take up a huge amount of space. These are chloroplasts, by the way, which we'll talk about in a second. There's a lot of functions of the vacuoles, and I'm just going to go through them with you one by one. Number one, it stores nutrients. Plants need to be able to have nutrients throughout the year, so we've got some storage area here. It does contain and degrade waste products, kind of like lysosomes, although plant cells do have lysosomes as well. They can accumulate poisonous materials. Think about this. Like, think about poison ivy, right? How does the plant cell not get poisoned by poison ivy? Well, it's actually holding it in these vacuoles. And so, yeah, there's some place where it's stored in the cell, vacuoles. They also contain pigments. For example, if you think about roses and being red, where is that red held inside the cells of that flower? And it's held inside of the vacuole. Also provides some physical support. It's like it'll press up against the cell wall. They kind of act as dueling forces, so also helps with support. That's your vacuole. The last one that we're going to talk about is the chloroplast. And with the chloroplast, what we have here is a structure that goes through photosynthesis. Remember that photosynthesis was first invented by the prokaryotes. Remember that chloroplasts were brought in through endosymbiosis. So this is basically a bacteria living inside a plant cell. And we're not going to go into any of the details here. We'll learn some of them when we get into ecology a little bit later in the semester. But for now, photosynthesis, making ATP, making sugar, that's what I want you to know about the chloroplasts. So this is just a summary slide of the major structures that we've talked about, whether they're in animal cells, plant cells, or both, and then what their basic functions are. Just as a quick review, you do need to know all of the major functions, but, well, there you go. So that's what I've got for you for Chapter 4. I think I might have made a mistake a couple times and called it Chapter 3, simply because the new textbook has different chapter numbers, and you can see some of the figures have Chapter 3, just because they have the same figures. So Chapter 4, this is the end of Chapter 4. Hope you enjoyed the lecture.